So this is the flow of the information using bioinformatic methods. So if we use, starting out with metabolites, the first step is to identify what the metabolites are, identify what they are, measure them, and then you think you have to know the information background to support the interpretation of all this data. You have thousands of different metabolites with a number or a level that you measure. So how do you make sense of it? So you have to rely on how they are related, what are the metabolic pathways that link these metabolites together. And then using the data, you do pathway reconstruction. Okay? From the pathway reconstruction, you know what is converted to what. Okay? And based on that, you do modeling and calculate where is the flow coming from, where does it start, which direction does it go. Okay? And then you do modeling and calculate the flux, the metabolic flux analysis. So measuring the data, a lot of times, would have to depend on what we know about what's inside the box. Okay? So if we do not know what's in the box, okay, we can only see the input, we can only see the output, and then based on that, we guess what's inside the box. Okay? And then based on from the guess, and then we generate new hypotheses and further investigation and do more research to figure out what's inside. Okay? If we already know what's inside the box, okay, then from measuring the input and measuring the output, we can model and see and tell what pathways, what happens when you do this, and then what happens to the individual pathways inside the box. So in order to figure out what's inside the box, we have to look at things in close detail. So it's like examining the plumbing in the bathroom instead of looking at the plumbing in the whole house. So to do that, the methodology, then we're not looking at like the whole genomic or transcriptomic type of analysis that analyze thousands and thousands of genes. We would be focusing on a panel or a selected number of genes that are relevant in a particular pathway based on prior knowledge. And that type of analysis of a metabolic pathway can be easily done with, for example, if you look at the RNA level, you can use the reverse transcription quantitative PCR method approach using the 96-well plates to analyze the targeted panel of genes. And Lim have done that. Lim was looking at the impact of the loss of 1453 sigma gene and the p53 in the glycolytic enzymes, expression of the glycolytic enzyme. So this is a focused panel of the glycolytic genes, enzymes involved in glycolysis, and using cells that are deficient in p53, or using cells that are knocked out without the 1453 sigma, and do the reverse transcription PCR analysis of the panel of genes, and you can see that the loss of the 1453 sigma and the loss of the 253 affect the different enzymes along the glycolytic pathway in different ways. So the same similar data can also be presented in a different way in terms of a heat map. So this is a transcriptomic data from measuring the metabolites of, for example, lysine, serine, alanine, uracil, all these metabolites from different cells are measured, and you can compare the differences in the change in a particular gene, the loss of a particular gene on the metabolite levels. So if you're focusing on a particular enzyme, okay, you can measure the enzyme. So you can measure the level of the gene that is being transcribed. You can measure the level of the protein expression, which is a balance between the translation of the protein and the degradation of the protein. You can measure the post-translational modification of the protein. Very often, phosphorylation is a very common post-translational modification. And very importantly, the activity of the enzyme can be measured by looking at the metabolites, the substrates and the reaction products. With single substrate reactions, we can model that the Michelson-Menten kinetics using applying the equation with the time course kinetic analysis. We can find out about the kinetics and the content, and calculate the kinetic constants for the reaction. So, for example, here, with the optical density, using optical density to measure the products, you can measure the rate of the reaction of a particular enzyme. Here, they're measuring the hexokinase activity. That you can see here that by changing the genes of the cells, by deleting p53 or deleting 1433 sigma, the activity of the hexokinase is affected to different degrees. ATP measurement is very important in the analysis of energy metabolism, and measuring ATP is, now these days people use the luminescence methodology, and basically when you lyse the cells, the ATP in the cells would become permeable and would be in contact with the luciferase enzyme that you put into the reaction mixture. And the luciferase would use the ATP to catalyze the reaction to oxidize luciferin to oxyluciferin. And in the process, light is emitted, and it's the measurement of the photon that quantitate the amount of ATP present. And for measurement of the mitochondria, to look at the mitochondrial potential, there's a methodology that uses the fluorescent dye. This dye is called the JC1. And this fluorescent dye has a characteristic that when the mitochondrial membrane is polarized, the JC1 is concentrated inside the mitochondria, and at high concentrations, they form aggregates. They form oligomers. These oligomers have a different fluorescence. They have a red fluorescence, whereas in the monomer form, they have a green fluorescence. So if you have the quantitate the ratio of the red fluorescence and the green fluorescence, and you can tell that the mitochondria are polarized. And from this ratio, you can measure, you're using the ratio of the red fluorescence and the green fluorescence, and you can run flow cytometry and have the, assess and evaluate the mitochondrial membrane potential of a population of cells. So here, Lin have done this experiment using this flow cytometry methodology and demonstrated that the mean fluorescence the mean ratio of the red to green fluorescence is changed when the 14-degree sigma gene is So, and there's another way to measure mitochondrial mass, meaning that you need to measure the number of mitochondria and the biological mass of the mitochondria rather than the membrane potential. And in that case, we basically just use a different dye. This dye do not form monomers. They're based on the membrane potential of mitochondria. They basically just stain or bind to the cardiolipin, which is the lipid content of the mitochondria. And with that binding, it sort of quantitate how much cardiolipin is present. And the fluorescence of this acridin, 10-nonil acridin orange dye would represent the biomass of the mitochondria. And here, Ling demonstrated that the loss of the 14-degree sigma would lead to a increase in the mitochondrial mass. So to look at glucose uptake, you can use a fluorescent dye that is conjugated you can use a glucose analog that is fluorescent. Okay, so this is a deoxyglucose analog, 2-NBDG. So this is taken up into the cancer cells through the glucose transporter, and you can see that here where the loss of the 14-degree sigma gene, these cells would take up more glucose compared to the wild type cells. So to measure hexone kinase activity, there's also the methodology of using absorbance. And this is a coupled reaction method. So if you look at this reaction, the glucose with ATP generated the product glucose 6-phosphate and ATP. None of these products have a significant absorbance and an impact on wavelength. But if we couple this glucose 6-phosphate with the another reaction with NAD, so the glucose 6-phosphate dehydrogenase, we can add this to the reaction mixture. And by fixing the concentration of the glucose 6-phosphate in the reaction mixture and provide a fixed concentration of the NAD, then we can catalyze this sequential reaction, the second step, to generate gluconate 6-phosphate and then ADP and NADH. Okay. And the NADH is the product that we can measure using the spectrophotometer to measure the absorbance at the wavelength of 340 nanometer. So for measurement of metabolites, the metabolomic approach can be used. And basically, these days we use liquid chromatography and mass spectroscopy or NMR spectroscopy. To measure signal metabolism, very often we can use radioactive substrates. To measure the glycolytic rate by the aldolase reaction, we can use the tritium labeled glucose at the number 5 position and look for a measure the amount of tritium labeled water that is produced in this reaction. If we want to measure the fatty acid beta oxidation. Then we can use substrate 910 tritiated pharmacy. Earlier, I already discussed about the handling of the metabolomic data, and there's a modeling that you can actually calculate the flux through the metabolic pathways. And they call this methodology called fluxomics. There's also in vitro culture of cells that you have this so-called metabolic analyzers that you can culture the cells in to actually measure the mitochondrial respiration as well as the glycolysis. For measurement of glycolysis, the machine is actually detecting in real time the production of protein in the culture media. And the machine also can in real time measure the consumption of oxygen. So in these in vitro experiments in these analyzers, you measure the OCR, the oxygen consumption rate, and the extracellular acidification rate, which is the proton secretion rate. And when you do that for a certain for 20 minutes for a period of time to get a stable reading, and then you add the drugs, oligomycin, and then followed by FCCP, antimycin, rotenone, etc. So the purpose is to assess what proportion is baseline production, what proportion is based on the oxidative phosphorylation, what portion is the spare respiratory capacity. So using these type of equipment, you can analyze in real time the impact of the loss of different genes on the metabolism in terms of glycolysis and oxidative phosphorylation. Now there's another methodology using the isotope labeling of the substrate. Okay, earlier I mentioned the labeling of substrate using the tritium. But you can also use the isotopes of carbon. You can use carbon-13, which is a stable isotope that's not radioactive, or use the C14, which is the radioactive. So depending on what your detection methodology is, if you're using radiation for detection by scintillation counting or by measurement of radioactive metabolites, then you use carbon-14. If you use lipid carbon field mass spectroscopy, then you use the stable isotope, C13. So with the stable isotopes, you can measure metabolic flux. So you can actually trace how much of the isotope label substrate is passed on to different metabolites in the cells. So when the cells are fed with a growth substrate of certain carbon positions labeled with C13, the distribution of the label in the intracellular metabolites can be calculated based on the known biochemistry of the plant. So there's also ability to look at the impact of the metabolism of different genes on the cancer cells, and then implant these modified cells into mice and look at it in vivo. So in vivo, we can use the PET scanning. Okay. So this is an example of a PET scanning. So this is a PET scan of a mouse. This is an MRI of the mouse showing that the actual size of the tumor that was implanted. Okay. So this is the same. So this is a different animal. Okay, that is a combo-sized tumor. Okay, but in this animal, the gene of the 14q sigma was induced using doxycycline. Okay, you feed the mouse the doxycycline induced the expression of the 14q sigma gene in the cancer cells. And when this cancer cell expressing the 14q sigma gene, and then you put these mice for the PET scan using F18-deoxyglucose, you can see that without the expression of the 14q sigma gene, the glucose uptake is very high. And then when you express the 14q sigma gene, it suppresses the glucose uptake in these cancer cells. In the mouse, the mouse is still alive. Okay, that's all, folks. It's been a long lecture. I think I've gone almost half an hour over time. Any questions?
Oh.
Sure, thank you for giving us the presentation. And I have some questions for you. First, I see that we have another enzyme for the glucopases, phosphorylfructokinase 2, PFK2. Does this enzyme present in normal cell or it will appear in the tumor cell?
PFK2.
Yes,
I'm not sure. Yeah, that's an analyzer of PFK2. I'm not sure whether the PFK2 is particularly the preferred form for cancer or not. So there are a couple of isotypes that are specially expressed in cancer along the pathway. One of the well-known ones is PKM2. That the M2 is in the cancer and M1 is in normal cells. And the LDHA is in cancer. And the LDHB not cancer cells. The PFK2, I do not know whether it is preferential in cancer or not.
Another question, sir. I see that the glucoses in the cancer cell has moved to turn to the lactate, but the cancer cell need more energy. But the pathway from the glucose to lactate just produce 2 ATP, but from the glucose to the pyruvates produce 36 ATPs. So how?
From glucose to pyruvate, 36 ATP?
Just 4.
Right.
But from the lactase, it produces less ATP.
Right,
So
because
how can
there's 2 ATPs getting used to convert from pyruvate to lactate.
But how can from the procedures that produce less ATP that will produce lots of ATP for the needs of the cancer cell?
Well, it's the flux. Okay. So, all right. Let me put an analogy. If let's say you paddle the gear of a bicycle. Okay? If you use high gear, you don't need to pedal that many times. Okay? But if you use low gear, okay, each turn of the gear you produce only 2 ATP. Okay? But if you pedal fast enough, okay, you pedal 100 times a minute, and then instead of pedaling 2 times a minute, then you're still generating more ATP. Okay? So that is why the cancer cells use a lot of glucose. Okay? So to generate, actually the cancer cells use more ATP than regular cells. Okay? So in order to meet that energy need, they need a lot more glucose, many fold, 50-fold, 100-fold more than the cancer cells.
And the last question is, why do we have to measure the mitochondrial membrane polarization?
When or how?
When and how?
When and how? Okay. So, well.